Hi, my name is Justin Wu, and I'm here to present my science computational model as a video since uh, my teacher was not able to review it in person. I'm in Dr. Pena's 1A class, so I'm uploading this there. Uh, my model describes the process of protein synthesis, which involves the transcription of DNA to RNA by RNA polymerase, and then the translation of that RNA to amino acids, which then create a protein in the ribosome. So let me explain that biological process. Since DNA is made up of a long string of nitrogenous bases, um, those nitrogenous bases are split up into genes, and when a cell wants to create a certain protein, it'll go find that gene, and then RNA polymerase will go there, and it will split up the DNA double helix, and then it will use one of those strands to use complementary base pairing and then create a complementary RNA strand to the DNA strand, and then I'll zip it back up. And complementary base pairing occurs in this way. For every A base, it'll change it into U base, G base to C base, T base to A base, and then C base to G base. The only difference between DNA and RNA is that instead of a T in the DNA, in RNA, it's a U, which is very similar, but it is a little different. Then the process goes like this. The RNA goes to the ribosome, and then it's translated by the ribosome in the codon system, which takes like triplets of the nitrogenous bases, and then the appropriate tRNA will come up and attach an amino acid to the polypeptide chain. And as you can see, that there are a lot of similar codons that output the same thing, but that's just the way the process goes. So yeah, this model describes the process from the DNA up until the polypeptide chain. So what variables does this define? The first variable it defines is base transcription dictionary, which defines a Python dictionary with the appropriate definitions for complementary base pairing like A to U, G to C, T to A, and then C to G. The second variable it defines is base translation dictionary, which defines codons to their appropriate amino acids, and all the possible codons are here. Then the third variable is codon strand in question, which is an empty string. Then the fourth variable is DNA bases, which is actually defined as a user input. I'll get to that later. Transcription yes, no is also defined as a user input. Codons deciphered, which will eventually be our result list, is defined as a list with a single element, which is a string that says there was at least one error in the translation process. Please try again. Def, start valid, def end valid. They're not technically variables, but I'm defining functions here. And then transcription yes, no is used again, but it is redefined with a new user input. And you can also technically call this a definition, but codons deciphered is redefined in this final section here as an empty list, as long as there are no errors earlier in the code. So the user inputs in my code are DNA bases and transcription yes, no, and there are no other user inputs in the code. Transcription yes, no is reused again, just as a same variable name with a new user input. But there are really only two variables that take in a user input. And what they do is DNA bases takes in the user's DNA base strand that they want to put through the protein synthesis process, and then transcription yes, no. In the first case, ask the user if they would like to see how the transcription process worked for that inputted base string. And then in the second case, it actually asks if the user would like to see the translation process. So the conditionals in my code, uh, I'll describe each one of them. The first one is on line 31. If transcription yes no is basically a yes, it checks for the cases y yes, uppercase y, and uppercase yes. Then it initiates this string, which prints out the original base in the input string, and then the transcripted base based on the dictionary, which it does this for each base in the inputted string, so it takes up a lot of lines. If the conditional is true, this happens. If it's not, then it just skips that, and it just goes on with the program. My second conditional is actually in a function. It's on line 40, so technically it runs a little later. But what it does is it checks if the first three um, characters of a inputted argument string, which in this case will be codon strand in question. Sorry. Uh, yeah, it'll it'll check if the first codon is methionine using the dictionary, and if it's not, then it'll do this error message, and then it'll prevent a polypeptide chain from being printed by the program. The same is true with the nvalid function. There's a conditional on line 46. It checks if the last codon is a stop codon, and if it's not, then it'll print this stop error, which is different. And then it'll stop polypeptide chain from being printed by the program. I have two more conditionals. One is on line 54. It checks if start valid is true and if end valid is true. So remember that start valid, if it's true, that means the first codon is methionine, and end valid, if that's true, that means the last codon is a stop codon. So if both of those are the case, then there should be no error in the process. And my last conditional on line 55 just asks, it uses the second version of transcription yes no. So if the user says yes, I would like to see the translation process, um, it will show the codon, and then it will also show what um, what amino acid that was turned into. So what does the loop do in my code? I have a few loops, 4 loops, I think. Four for loops. So the number 4 and then four for loops. So the first for loop is on line 32, and like I mentioned, it for I in range length of DNA bases, and DNA bases was the input string with the DNA bases. So for each character in that string, which each character would be a base, it'll print that. It'll print this text arrow, and then it will print based on the dictionary and based on complementary base pairing what that base was turned into, and then what it was appended, like what was appended to the RNA string. And the next for loop does something very similar. It appends the it's not really an append because it is codon strand in question as a string. It's not a list. So this isn't really an append. It's a concatenation, but it concatenates the base that was transcripted using complementary base pairing, and it adds that to the end of codon strand in question. And it does this for I in range length DNA bases based on how many bases there were. Then my last two for loops here are they do a similar thing, but for the translation process, so they're a little different. They use for I in range 0 length codon strand in question, and then 3, this implementation of the range function of the for loop means that it'll start at 0, index 0. It'll go up to the length of codon strand in question, but then it will skip by 3. And what that does is it goes to the start of each codon, and that's really important because we're actually measuring the index we're at, the index to the right of that, and then the index two spaces to the right of that. So that's why we need to skip by 3, and then it will add um, the correct anticodon and amino acid based on the translation dictionary variable. And then the next for loop does something very similar, but instead of printing it for the user to see, it will append it to codons deciphered, which is our result list. So my code does use an additional technique. The additional technique it uses is lists, and it also uses functions. Um, the one list used in this code is codons deciphered. It'll be a little easier to see why it is a list once we run the program, but it is initiated as a list with one element. And then in this last part of the code, in lines 54 to 60, we use the append function to utilize that list effectively. Um, in terms of functions, I think they were really important for this because start valid and end valid, they help see whether or not translation could even happen, and if the inputted DNA strand is valid, because start valid and end valid are both true because of the things they measure, which is whether or not the inputted DNA strand starts with methionine and ends with a stop. If one of those is not true, then there's going to be some error in this protein synthesis. So that it adds to the program because it shows whether or not there would be a biological error present. And it's important that they were functions because it's difficult to put this code here without using a function. Okay, then I'm now going to run my code twice using different values on it. I have my values preset here, so I'll just copy them and run the code. So it asks enter DNA bases of it in a gene below. So I'll add this, and for the purposes of this program demonstration, both of these values work good. So yes, I would like to see the transcription process, and it will show me the transcription process. It'll show me that T was converted to A, since that's the first base in my input string. A was converted to U, and at the end T was converted to A, so this T would be converted to this A. So yes, I would like to see the translation process, and it will show me how the translation process worked. So AUG, which can be seen here, AUG, yeah, it recognizes that as methionine, and then at the end UAA, it recognizes that as a stop codon. So it says amino acids, and then it prints me a list with the amino acids, which would represent the polypeptide chain. So let's rerun this code with the second preset here. It's a little longer, and there are some differences. So yes, I would like to see the transcription process, and it shows me the transcription process same as before. And yes, I would like to see the translation process, and it will show me the translation process. And it'll show me it starts with AUG, but it does end with AUG up here, sorry, and then it ends with UAG, which is a different stop codon. I'm going to run my code one more time to display how the error messages work. So here, if I start my code with something like, instead of TAC, I start it with TAG, and then I end it with a valid end, like ATT, which would produce UAA. It will show me the transcription process, but in the translation process, it'll print me the error message I set up, which says the inputted string does not start with methionine. Consequently, a real translation sequence would execute incorrectly by omitting the first few codons. And instead of printing me the polypeptide chain, it'll give me the error message. There was at least one error in the process, and it'll tell me to please try again. Okay, thank you.